Hello friends, welcome to BMH Learning. In this video, we will talk about sucrose density gradient centrifugation. This technique is divided into three steps. First is preparation of sucrose gradient. Second step is centrifugation. And finally, separation and elution. Let's start with the first part, the preparation of sucrose gradient. As the name suggests, the technique requires sucrose, which is nothing but a disaccharide formed of glucose and fructose. So initially we make a solution with 66% sucrose. The key point in this centrifugation method is that the sucrose is to be used in different concentrations. So for that, the most concentrated sucrose solution is diluted to various desired concentrations. Now, how does a sucrose gradient look like? So we have sucrose density gradient column, and this column has sucrose. But the basic idea is that the sucrose is not uniformly distributed, but instead it's arranged in different layers, such that the layer on the top of the tube has very low density sucrose. On the other hand, at the bottom we have very high density sucrose. So the layer at the bottom is very dense. And in the middle, you have all different densities of sucrose, which are gradually getting dense as you come down. So we can say this layer is having intermediate density, which is more than the top and lower than the bottom. It should be noted that when adding the higher concentration of sucrose solution, the tip of the pipette should be placed at the bottom of the gradient column. You must also avoid disturbing the interface between the layers. The prepared gradient should be used as soon as possible to avoid premature diffusion of the gradient layers. Now let's move to the centrifugation step. So we have the sucrose density column having different densities of sucrose. What we are going to do is that a suspension of the particles is layered over the top of the solution in the column. And then we put the sucrose density column in a centrifuge machine. So what would happen? The centrifuge is going to spin the column, and the centrifugal force exerted by the spinning of the centrifugal tube will cause the separation of the particles. Basically, what happens is that the proteins in the suspension will move into the sucrose gradient, and the density of the different protein molecules in the suspension will determine where the protein will end up in the gradient column. To understand this, let's say the suspension we used had 3 different proteins. Suppose the orange protein is the one having least density, and the blue colored protein is the most dense, while green one is of intermediate density. So, upon centrifugation, the protein molecules would go into the centrifuge tube, and a particular protein would migrate up to that position in the sucrose gradient, which is having the similar density to the protein particle in question. It's because the higher density layer would not allow the lower density molecule to get into it. So we will end up like this. Different proteins arranged in different layers of sucrose according to their densities. After centrifugation, particles obtained from different layers or bands need to be further separated and purified. The separation of the particles can be done in 2 ways. With the help of instruments like gradient fractionator, different components can be separated and collected in a bottom to up fashion. Or by using a pipette, they can be separated and collected from top to down. After we are done with the separation, the fraction we get is a mixture of both the solution and the particles we need. Now what we require is the elution to further purify the fraction. This was all. Thanks for watching.